So we're going to review our cell scientists. On Wednesday, last week, we did the fabulous convention of cell scientists. And what we had was these 7 scientists, and we talked about what each one of them contributed to cell theory. So let's take a look at each one, and you can add to your notes. If you don't have your notes, I guess you can start fresh, but you should have a little something about each one. So these are in order, in chronological order, that these things occurred. I don't care that you remember exactly the names, but as you see as we go through them, by knowing them roughly in order, it helps kind of put perspective on what was going on. So Robert Hooke. Any of those Hooke people out there want to tell me what Robert Hooke did? Everybody forgot by now? Yes. Okay, excellent. He was English, and he saw the very first cells, and yeah, he discovered dead cells, which might seem a little strange, but the way this all happened, he was using, it was basically a child's telescope that he was playing with that he had gotten as, he had bought it somewhere, and he thought, well, this is interesting. What else can I do with this? And so he took this and started looking at things because it had good lenses in it. And so with this child's telescope, he started looking at different things, including a very thin section of bark. This is actually a recreation of what he drew. This is what he saw when he looked at a thin slice of bark. And back then, of course, this was in the 1600s, there was no camera, so he had to draw what he saw. And so he saw this little webbing kind of stuff like this. And as you guys looked at your ponds, you probably saw things like this, dead things. And so he thought, oh my goodness, this piece of cork bark is made up of little empty rooms. And so he called them cells. So this was in 1660, and he actually came up with the word cells, meaning like a little empty room. Okay? So that was Robert Hooke. He was English. Again, I'm not going to have you memorize what everybody's nationality was, but the point of all of this is that it takes people from all over Europe to come up with what we're going to have with cell theory. So scientists then, just like scientists now, are very competitive. And so he goes to his scientist meetings, and a lot of these people weren't full-time scientists. They did science in their spare time for fun. And so he went to his scientist meeting and said, hey, everybody, guess what I did? I took these really good lenses, and I was using them to look at things. And look, this wood, this cork bark, is all full of little bitty rooms. And so everyone's like, oh, well, that's kind of cool. Let's see what else we can look at. And so our next researchers who we're going to hear about were looking at other things. Now, here's one of my favorite people, Anton von Leeuwenhoek, who was Dutch. Who can tell me what von Leeuwenhoek discovered? Von Leeuwenhoek's out there? Yes. And this is one of my favorite ones too. He was looking at pond water. And so he actually saw the first living cells. And so this is about the same time, 1674. So that says that's about 15 years later. But you see this is going on and on. Saw the first living cells in pond water, very much like what you guys are seeing. So think about that. He's looking at pond water and he sees little things swimming around. Kind of creepy, right? He called them animalcules, right? So instead of molecules, animalcules. I don't know why we don't still use the word animalcules. I think it's kind of a nice word. He also looked at teeth scrapings, like if you scrape the little plaque stuff off your teeth. He scraped off that and looked at that under a microscope. He looked at everything he could think of under a microscope. You know, bits of food, bits of skin, bits of human tissue. He looked at everything he could think of under the microscope. But he did see the first living cells. He also saw the first bacteria, which are much, much smaller, which is what was in the plaque off of his teeth. It's kind of gross. But that's Anton von Leeuwenhoek. The way I remember these guys, Hooke, of all the people we talked about, Hooke really has the simplest name, right? Hooke. And guess what? He discovered the simplest thing. So Hooke, simple things. Von Leeuwenhoek, big, long, funny word. He came up with animalcules, kind of a funny word. More complicated, So Hooke, von Leeuwenhoek. Hooke follows von Leeuwenhoek. Okay? Kind of help you? All right, then next we're going to have these three scientists all together. We're going to have Schleiden. So this started this whole thing of everybody was studying things. And Schleiden, who was, are these three guys, these next three, Schleiden, Schwann, and Virchow, they are German. All three of them are German. So Schleiden did lots of study, and he decided to study plants. After much study, every plant he looked at was made of cells. So after a while, he said, you know what? I think all plants are made of cells, as in about 1838. So we're almost, 150 years later. All plants are made of cells. So this is just something that's ongoing. It's the big thing that people are studying. And about the same time, Schwann, after much study, stated, you know what? All animals also seem to be made of cells, right? Every animal he could think of, he's testing the tissues he's looking at. All animals are made of cells. This is all about the same time, okay? And then a man named Virchow, sounds more Russian to me, but he apparently was also German. He came up with cells only can come from other cells. And he actually, under his microscope, saw cells dividing. So he saw that a cell could make another cell. And he said, hey, I think that cells really can only come from other cells. And in just a second, we're going to talk about why that's actually kind of important. But first, how do we remember Schleiden and Schwann? Schleiden and Schwann. How do you remember who did plants, who did animals? Yes. Yes. That's the way I remember. Schwann sounds like swan. A swan's an animal, right? And so I just remember Schleiden and Schwann were plants and animals. Swan was obviously the animal. And after our first period convention, I got better at giving the directions, and the kids got better at coming up with what was Swan's favorite animal? A swan. Schwann's favorite animal, a swan. What was Hooke's favorite color? Brown, because he was looking at bark. And so we were doing silly things like that to help you remember. And one group said, Schleiden means to be quiet, and plants are quiet. Okay, if that helps you remember, then that's awesome. And then Virchow, I don't know how you remember Virchow, but cells only come from other cells. Okay? So those are those five, and then we're going to talk real quickly about the other two and what they had to bring to it. Questions about these guys? Okay, so next we have Redi and Pasteur. And in the textbook, it talks about something called spontaneous generation. Did anyone understand what spontaneous generation was that these guys were disproving? Yes. Yes. Now, this seems really strange to us, but spontaneous generation was the idea, the observation. So spontaneous just means kind of like out of nowhere. And generation is life, right? Having a new generation of life. So life coming out of nowhere. Now, this might seem preposterous to you and me, but from what they observed, have you ever left some fruit out on the countertop and then all of a sudden there's these little fruit flies that just came out of nowhere? Well, they thought, well, the fruit flies must have just come out of the fruit. The fruit must have made fruit flies. Or if you have a compost pile with some nice soil and rotting food and plants, that would create worms and flies. Because that's what they observed to happen. They didn't see the microscopic things, and so they observed, incorrectly, of course, that life came from non-living. And if you look, if you, look it up a little bit, there's stories too, like if you leave a pile of rags with some rice and crumbs, mice will grow out of it. You can imagine how they might observe that, but we know that the mice didn't come out of the cloth and rice. What's that? A very rapid reproduction. That's right. Just almost magically from nowhere. Okay, so then we had these two scientists who who came up with really important experiments that disproved spontaneous generation, which is, and coming into why cell theory ends up being really important. So Francesco Redi, who had Francesco Redi or knows what Francesco did? Anybody else? All right. Okay, excellent. And it's really interesting. You'll learn about this same guy in high school or college, and his experiment was actually a lot more sophisticated even than just what your book says. But so the thought was if you leave meat out, maggots will grow out of it. Because what people were not seeing is the fly would land on it and lay eggs that were microscopic. Those eggs would grow into maggots, which grow into flies. But since people weren't seeing that step, they thought it grew out of the meat. So I remember Redi, because he did this kind of disgusting experiment with red meat, but he had one of the first very controlled experiments. So he actually had one jar that was open to the air so the flies could land on it, and he had another jar, and he couldn't just close up the top because then people said, oh, well, maybe it needed air. So he put a cloth over the top that was fine enough that, the flies couldn't lay their eggs through it. And the one that was open to the air grew maggots, and this one did not. And so the things he did was, one is showing that those maggots had come from some other life, and the other one was having a repeatable, controlled experiment. So that other people, if they say, no, 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 we've believed this forever, flies come out of meat, he could actually show them, here's an experiment you can do yourself. I can show you the steps. Okay? So those are the two really important things from Redi. And then Pasteur actually does something similar. And again, Redi, red meat. And so people were saying his favorite color was red. Redi was ready to experiment. Anyway, Pasteur. What did Louis Pasteur do? So Francesco Redi is Italian, Louis Pasteur is French, and so once again, people from all over Europe contributing to this over the years. Louis Pasteur, anybody? All right. Louis Pasteur did a similar experiment with broth. Think of chicken broth. Oops. So like chicken broth, and so he had these funny looking flasks, but basically if you leave your chicken broth out, it will grow disgusting, nasty mold in it, right? If you just leave it out, right? Chicken broth. Yeah, but when it goes bad, boy, does it smell bad. And so he wanted to do something similar. So to make it so the air could get in, but there's no bacteria in it, what he did was he put it in this funny container like this. And then what he did is he boiled it. Why did he boil it? Yeah. Yeah, he boiled it to kill all the bacteria, to sterilize it. And once he had boiled it in this little container, it stayed fresh. And then if he broke this little glass thing off, and then the air, just the regular air has bacteria and mold and stuff in it, then the mold would grow again. So again, it was a very famous experiment. You can imagine this is really helpful to know this stuff, to know that diseases are transmitted this way, food poisoning, all this kind of stuff. So Pasteur, have you guys ever heard of pasteurization? Pasteurized milk? Yeah. The milk we drink, and actually many of the fresh juices and stuff are pasteurized, which means they have this quick flash heat sterilization so that you can keep them even in your refrigerator for a week or two before they start going bad. Before that, your fresh fruit juices or milks or whatever would go bad much more quickly. Okay, so pasteurization. He also later came up with the rabies vaccine and stuff like that. Okay, those are our 7-cell scientists. Everybody good with 7-cell scientists? Now, the last thing we need to know. Oh, does anyone have what year these guys were doing this? In the 1600s? So this is going on about the same time as all this other stuff. So then we have cell theory in 1855. So we started out in 1600 with the very first microscope, or 1660, all the way to 1855. So this takes almost 200 years to come up with this. Science moves very slowly. But you can change science if you have well-organized experiments. So this should be no big deal. All living things are made of cells, right? So that comes from what's his name? Von Leeuwenhoek. It comes from Schleiden and Schwann. All living things are made of cells. Now this one I think is a little confusing. Cells are the basic unit and structure and basic unit of structure and function in all living things. Well, what does structure mean? What it's made of, what it's built of. And what does function mean? What is the function of something? Yes. Yeah, how it works. What job it does. So cells not only build us, but cells help us work properly. So in order to breathe in air on a cellular level, you have muscles contracting to move your muscles in your lungs, and you have oxygen going into the cells. So cells don't just make up our structure, but they also make us work. And then we have all cells come from other cells, which was, of course, so Redi, Pasteur, Virchow. And this one here is, of course, Hooke, von Leeuwenhoek, Schleiden, Schwann. And so you can see how all of these people over all of this time, sorry about that. Okay, all of these people over all of this time comes together, and over 200 years, we now have cell theory, which again seems pretty obvious to us today. All living things are made of cells. Okay, any questions? Check this out. Where have you heard this before? All living things are made of cells. It's one of the 6 characteristics of all living things. Oh, yeah. All right, any questions about any of that?